Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Well, welcome everybody back to In the Interim. I'm your host, Scott Barry. And as you can tell, my studio has changed. So the new studio, which if you go back a bit, seasonally, that I am here in northern Minnesota, and you can see beautiful. That's actually that's not a virtual background to that. That is the woods outside of our little cabin here in Minnesota. We get to escape the heat of Austin, Texas, and spend some time here in the woods of Minnesota. So I'm in a little office above the garage here in northern Minnesota, and I appreciate you joining me. So I want to introduce today's topic with a bit of a panicked email and then phone call I got in October 2016. The panic was from my father, my collaborator on I Spy 2. And you can go back to previous episodes of In the Interim looking at the I Spy 2 trial. We actually did 2 consecutive episodes about the trial. Fantastic trial. I'll introduce a little bit, but it is an important part of the story is the primary endpoint in the trial. The trial is I Spy 2, neoadjuvant breast cancer. And the important part of the neoadjuvant part of this is that what that means is rather than traditionally surgery, you're diagnosed with breast cancer, a surgeon goes in and removes as much as the breast cancer as they can, and then you're given therapies. That's adjuvant treatment. Neoadjuvant is where you don't go in and remove the tumor immediately. You give treatment. And 6 months later, surgery is done. And an advantage of that is you get to see whether or not the treatment had any benefit on the tumor itself. The primary endpoint in I Spy 2 was pathologic complete response. That means that when surgery is done 6 months after the start of the trial for a patient, is that the cancer's gone. It's a complete response. There's no evidence of tumor at the time of surgery. And again, this is the statistician telling you the endpoint. An oncologist, breast cancer oncologist, could do a much better job of this, but I'll give you the statistical needed parts of this. So that surgery is done 6 months after and there's no evidence of cancer. The tumor's gone. Meaning that the treatment that you started and initiated 6 months earlier had really strong benefit for that patient. A lack of pathologic complete response would be that surgery is done and there's tumor remaining that the surgeon removes at that time. Now, that endpoint is a 6-month endpoint. In the trial, we don't know the result of that endpoint for a patient for 6 months. Now, in the trial, there are earlier readings within a patient that might be predictive of that response. There's an MRI that's taken 1 month, 3 months, and then actually before surgery. We call it 6 months, but it's relatively proximal to when surgery is done. And that MRI gives an estimate of the amount of tumor that is there. And now we'll go back, and even at 6 months, for example, the MRI might say there's no tumor left, but surgery reveals some layer, some aspects of tumor. So even at 6 months, it's not perfect. Now, in the trial, those values, the MRI values, are used within a patient in the modeling. I'll come back to that. What is that? What is the email and phone call panic that I got? Is that pembrolizumab, which was one of the treatments in the trial, had triggered graduation. Graduation is an adaptive outcome in the trial where there's at least an 85% probability that treatment would beat control in a phase 3 trial of pathologic complete response. Now, it's uncertain the role of pathologic complete response, and do we need disease-free survival as an endpoint? Do we need a different endpoint in this? And what's the role of pathologic complete response in breast cancer? Let's not get into that. But in the trial, that's one of the outcomes. And think of that as a positive outcome that is graduated. It means enrollment in I-SPY stops. All patients would go through 6 months, they would continue on their therapy, but no new enrollment would be given. Essentially, the treatment has had a positive outcome. I-SPY 2 is a platform trial with many therapies going through it, pembrolizumab being one of those. Now, the panic was, and I'll back up even a little bit more, is that in I-SPY 2, Berry Consultants wrote the code that runs that adaptive trial. The trial uses response adaptive randomization across multiple subtypes of disease, hormone receptor status, HER2 status, and the adaptive design was driven by Bayesian modeling of that. And Berry Consultants and I was involved in writing the code for that. Now, coming back to that, what's the panic? At the time that it triggered for graduation, the estimated pathologic complete response rate for pembrolizumab was 60%. And the control rate had an estimate of 23%. Very, very successful in the trial. The panic was only one patient at that time had an observed outcome of pathologic complete response, had the 6-month surgery, and we knew the data on one patient out of the patients on pembrolizumab. And the panic was, what's happening? Is the modeling broken? We have one patient, and it estimates a 60% response rate compared to the 23% on control. Something must be broken. Okay, so that's the panic. Now, I'll come back to that part of the story, but I want to shift things to adaptive designs. And I-SPY 2 was an adaptive trial. And I'll come back and talk a little bit more about I-SPY 2 in this October 2016 panic from I-SPY 2. But let's talk a little bit about adaptive trials. So what is an adaptive trial? An adaptive trial is a clinical trial that has pre-specified dynamic aspects to it. I try to not use the word change because the protocol is set up to have these dynamic pieces to it. So in I-SPY 2, there's adaptive allocation to the different treatment arms. There's triggering of graduation of an arm. All of that's pre-specified. The protocol doesn't change. It says in the protocol these things will happen. These are key aspects of the trial that are dynamic and by design can happen in the trial. Yes, from the perspective of the design, it feels like these are things that are changing in the trial. We stop enrolling to pembrolizumab when it hits a trigger of graduation. The role of that is to accelerate development of it to get it to phase 3. I-SPY 2 is a phase 2 trial. Now, during the trial, these things are triggered by data in the trial itself. Now, it seems, when I explain to people one of the things I do as a statistician, the main thing Barry does is build adaptive trials. And I've been building adaptive trials for 26 plus years now. It seems strange that, okay, I don't understand. You mean normally the trial doesn't change? And to explain that is really the opposite of an adaptive design, is a fixed trial. We at the beginning of the trial, in a fixed trial, you say, let's enroll 200 patients on a control, let's enroll 200 patients on a treatment, and let's wait till all of those patients get 6-month data, and then we'll look at the data and figure out whether pembrolizumab or whatever treatment it is works. And then we make the decisions going forward. That's a fixed trial. Now, many times in a fixed trial, you look at the data and you say, oh, shoot, I wish the sample size was smaller. We might have known this treatment was effective long before we got to 200 on each arm. In I-SPY 2, it's 120 is the maximum. So long before you get to 120, you might think that treatment works or it doesn't work. Or I wish we'd have put different kind of patients on that treatment compared to another treatment. I wish we would have changed doses. Well, generally larger outside of I-SPY 2, we could be changing doses in a trial. We could be altering the patients enrolled in a trial. We could move it to a different stage of trial where it selects a dose and it moves to a confirmatory part of the trial. And I'll talk about a trial that did that today. So lots of things in the trial can be dynamic. Now, why is that? What's the promise of adaptive trials? We think about fixed trials where you say, let's go enroll 200 patients on each arm and we'll look at the data when all of the patients have reached a time point that we consider primary. We look at the data and we learn something from that, and maybe we approve a treatment, maybe we go to phase 3, maybe we run another phase 2. Whatever it is, the role of the trial, that's a fixed trial. The promise of this is that we can change this trial in ways that improve the trial by changing the allocation, by changing the doses, by accelerating to another stage. We make the trial design better. That's really promising. There are a lot of things in trials, when in a fixed trial, we guess at. We guess at the effect size. We guess at which patients might benefit from it. We guess at the doses. We guess at a lot in the trial. We guess at the variability of the outcome, the prevalence of events in trials. We guess at a lot. The promise of adaptive trials is during the course of the trial, you learn a lot about that. If you look at the data in the trial itself, you learn how effective the treatment is. You learn about variability. You learn about the behavior of different treatments, the behavior of different doses. And then we make the trial better given that. Had we known that information before the trial started, we would have designed a better trial. That's the promise of adaptive trials, that we learn things to make that trial more efficient. We make the engine better. It's an incredibly promising thing of adaptive trials. if you think about it, the opposite of it is you close your eyes and you hope everything you designed 4 years later was the right thing to do. So that's the promise of an adaptive trial. Now, the crux of it is, as that adaptive trial is going, we need mechanisms to learn. There's data, and data's being generated in the trial. We need to learn from that data. If you think about I Spy 2, we need to learn from the pathologic complete response outcomes that come out of that trial at 6 months. In an Alzheimer's trial, we might be interested in the 18-month response. In a trial of a vaccine, we're interested in events that are somebody comes up with the disease that the vaccine's supposed to combat. And we're waiting for events to happen in that. That becomes the thing we're learning from. So we need models in the trial itself as a learning mechanism. And those models drive the dynamic things in the trial. That's what we need. So there's two key parts to an adaptive trial. We need to learn so that we make changes. And then when we design the trial, we figure out what changes in the trial make learning more efficient, make the trial better, treat patients better in the trial. All of those things are the dynamic changes, but we need the data in the trial to learn. If we try to make adaptations and we've learned nothing in the trial, it's all based on things we thought going in, which is we already designed the trial as well as we can for that. So it's really these two key things. We need to learn from the data, and that drives changes in the trial that make it more efficient, make it a better trial. So in that I Spy 2 example, graduation is an adaptation in the trial. We need to learn from the data to say, this treatment should graduate. Now, in that circumstance of the panicked email we have one patient at 6 months. The patient was a positive response. It was a pathologic complete response, but that's one. How is your learning mechanism saying we have at least an 85% chance that it's better than a 23% response rate? If it's based on 1 out of 1, we're not going to come up with an 85% probability. The data is not strong enough. In that trial, we have built models to accelerate the learning. Now, you come back to this whole question about adaptive trial. If we're waiting for everybody to get to 6 months, the trial's slower. The trial to make changes, to make dynamic adaptations, has to wait a long time, has to wait for patients to get to 6 months. Meanwhile, we've enrolled a bunch of patients earlier than that. Have we allocated them correctly? Now, in the trial, I brought up these MRIs that are done at 1 month and 3 months. In I Spy 2, we built a model that looks at the correlation between the MRI, which is a quantitative measure of the reduction in tumor from baseline. 100% reduction is getting to pathologic complete response at 1 month and 3 months. If there's no tumor in the MRI, we think that patient is highly likely to be a pathologic complete response. And hence, patients that have taken pembrolizumab that have MRI values that are really positive or really negative inform what we think the pathologic complete response is through what I'm going to call a longitudinal model. So the longitudinal model is the statistical mechanism that's learning what does 1 month MRI predict about 6 month PCR? What does 3 months MRI predict about 6-month PCR? What does 6-month MRI predict about 6-month pathologic complete response? Surgery happens after that, not too far from it. So it's not a huge benefit to the trial at 6 months, but 3 months can speed up the learning. 1 month can speed up the learning. So we build models for that to accelerate the learning about the primary endpoint. We haven't changed the endpoint. The endpoint is 6-month pathologic complete response, but early values are driving that estimate, which are driving the adaptation. Now, an important part of, what does that model look like? And I'll come back to specifically what that model looks like and how we learn from that. That's the aspect in adaptive trials. So when people ask me what scenarios, what trials can benefit from adaptive design, the most important part of it is that speed of learning. So if you have a trial where the endpoint is a migraine trial, where it's a treatment of acute migraine, there are treatments that are trying to prevent migraines, but suppose it's the treatment of acute migraine. A lot of times we look at 2 hours later, are you pain-free? So when you give a treatment to a patient, you know the outcome of that patient 2 hours later. We can do a lot of really efficient things adaptively because the learning ratio is really fast. And so we can be very efficient. Alternatively, if the endpoint doesn't come for 18 months in that scenario, we've got a whole lot of patients that have been enrolled that we don't know anything about, and we're waiting for them to get to 18 months. It's hard for the trial to be efficient. Should we stop enrolling? Should we change doses? Should we make changes? We don't know anything at that point. So can we speed that up? So adaptive trials become more efficient relative to fixed trials the faster we can accelerate that learning. And that becomes a huge part of adaptive trials. And it becomes the huge fight that we have with time. We're fighting time in that case to make the trial more efficient, to treat patients better, to learn more efficiently. That's the whole promise of adaptive trials. A huge part of building adaptive trials is this acceleration of learning within single patients that are moving through. It's what I've been doing for 26 years. Now, the interesting thing about it, a lot of fixed trials don't care about this. If your primary endpoint is 12 months and you're going to enroll 200 patients and you're going to wait till everybody gets to 12 months, you don't really care if 6 months is predictive of 12, if 3 months is predictive. It's irrelevant to the primary analysis. Now, you might do an MMRM model that incorporates that, but it's all done when all patients have had the opportunity to complete the primary endpoint time. Now, the whole world of this is missing data, but this is different kind of missing data. These are patients that just haven't reached that time point, which is a whole new thing in adaptive trials that don't come into it in fixed trials. So this whole thing of trying to accelerate the learning is new and relatively specific to adaptive trials. Okay, so a lot of what we do is spending this time on building that, predictive of it, in it. So thinking about fighting time here, let's go back to I-SPY 2. So in I-SPY 2, what does this model look like? At 1 month and 3 months and 6 months, we get an observation of the size of the tumor relative to baseline. And let's think about percent reduction. So the endpoint that we get at 1 month and 3 months is a percent reduction. we could get 100% reduction. We could get 50% reduction in that. Now we're interested in, suppose somebody at 1 month, a woman at 1 month has an 80% reduction in tumor size. What does that mean about the likelihood of pathologic complete response? That's really what we're interested in from that perspective. The beauty of the trial itself is that patients pass through 1 month and 6 months. We get to see a lot of observations on women that went from 1 month and had an MRI, and then we found out, were they a pathologic complete response? So we get a lot of pairs of what was the percent reduction at 1 month, and were they a responder at 6 months? So we're building a model here for that prediction. You could build a logistic regression model for that as that percent reduction to a response, yes, no. Now we built that original model, and a lot of times we're building that model in adaptive trial, and we don't have the data in the middle of the trial. We don't get the luxury of opening up the data and building a model and saying, oh, this is what the behavior of the model, this is the best fitting model, here's the BIC, whatever modeling approach you take. We don't have it. We build it before the trial starts. It's hard coded. It runs the model at that time point. So we have to build the model ahead of time. We had the luxury in I-SPY 2 of actually I-SPY 1. I-SPY 1 was an observational cohort. It didn't change the treatment of patients, but it got these observations. And so we had about 40 patients where we could build a model. Now, we want that model to be robust. We don't want it to be overly strong in ways that it's making bad forecasts of PCR, because it's somewhat hard-coded and it's running that, and it doesn't necessarily know it's not fitting well, and then it's driving decisions about allocation to women that with breast cancer based on a model that maybe isn't fitting very well. So we want this to be really robust. So the model we built in that circumstance is a piecewise prediction model where we actually had 13 pieces to the model that then forecasts the relative odds of a PCR. What do I mean by that? We broke that interval up into, suppose the reduction is 0 to 10%. That's an outcome. Oh, by the way, the tumor could grow. That's the worst outcome. Then it could be at 0 to 10, 10 to 20, 20 to 30, 30 to 40, and so on, all the way down up to 80 to 90. And then we break 90 to 95. Remember, pathologic complete response is 6 months, there's 0 tumor. So it's 90 to 95, 95 to 99, and then more than 99. Those are the 13 classifications of the MRI at 1 month. And then a separate instance of the model is at 3 months, and a separate instance is at 6 months. Then we learn that the relative odds of a responder as a function of which category your MRI is in. And based on that, that's learned from past women in the trial. It's one of the beautiful things about a platform trial is we have a lot of data on that in I-SPY 2 from previous arms and the control throughout that. So that model is running. So at the time, let's go back to this time where one patient had gone through, and this estimate was 60%. We had a number of women at that time in the trial And I'm going to try to remember, I believe we had some 60 plus women who were on the arm that had been allocated at that time, but only one had passed through. Now, all of the other women in the trial have some level of maturity of their MRI data, 1 month, 3 month. Some haven't even reached 1 month, and they don't inform the response, the PCR 6-month response. And then we have some that have 6 months but haven't had surgery yet at that time point. So when this panic happened, of course, we jumped in and we're looking at the model. You want humans to watch this automatic pilot to make sure that if the model is broken to some extent, that we make sure it's doing what it's supposed to do. So we went in and we investigated. And as Don says, pembrolizumab was just melting the tumor away. The values of the MRI for that treatment were really, really good. Lots of them were 99% reduction, 95 to 99% reduction. That the model was predicting that a lot of these were going to be 6-month PCRs. And the fraction of them that are going to be 6 months PCRs was 60% out of that. It was all based on the longitudinal model. We had one patient. It was entirely driven by the longitudinal prediction of those patients. By the way, at that time, I should back up. Some 60 patients had been enrolled with pembrolizumab. The I-SPY 2 trial had these different categories, different subtypes that are differentiated largely by HER2 status and hormone receptor status. One of those subtypes is called triple negative breast cancer, and that's that you're negative on hormone receptor status and HER2 status. There were 29 of those patients that had been enrolled, and one of those had passed through the 6 months. That was the subtype that was estimated to be a 60% response. At the time that it said, graduate this, stop enrolling, and pembrolizumab should go to phase 3. The final analysis takes place when all patients get to 6 months. It's another really, as statisticians, it's just another really neat, exciting area where we're doing these forecasts. It's forecasts and predictions of what's going to happen. And you stop enrolling a treatment arm, and then you get to see the outcome of it. So you're doing these predictions, but you get to observe the outcome. In many circumstances, we might make predictions and we never actually get to see the outcome of that, but it's forecasting what would happen. Here we get to see the outcome of that. So there was nothing wrong with the model. It was functioning exactly as designed. Made some people nervous that it was this forecast entirely. Well, the pembrolizumab stopped enrolling, and the trial read out, and it was published in JAMA Oncology. And the final estimate for the PCR response rate was 60%. It actually, the final posterior estimate of that, it triggered it almost exactly in the setting. Really, and by the way, it was an over 99% probability of winning phase 3 trial in triple negative breast cancer. And of course, pembrolizumab is an amazing therapy now in many, many different cancers. So it actually hit exactly. Ended up 60% to a control rate of 22%. And you can go see the posterior distribution in that paper, Nanda et al., and within that. So it triggered it perfectly 6 months ahead of what if we'd have waited for all of those patients. And meanwhile, we would have enrolled more on that. Now, in the design, you want to set up, what do we want the trial to do? Do we want it to do that? These are all decisions made before the arm started in the trial. Okay, so accelerating it, that's a case where even 6 months, by the way, a huge problem in breast cancer is there are really many effective therapies looking at endpoints like overall survival, event-free survival. These endpoints are thankfully very, very long. To do trials and learning trials in an adjuvant setting looking at those endpoints are really, really hard and long. And so a huge desire in many of these diseases are looking at endpoints that are earlier, that are predictive. That's a different part of disease modeling and longitudinal modeling, hugely important. And that's a separate thing. Look for surrogate markers and huge thing that Barry does a lot of that modeling in other diseases and cancer. But this is the setting where let's sort of fix in that the trial is a primary endpoint. And within that trial, we're trying to accelerate to that primary endpoint. Okay. What are other examples of this? What are other examples of fighting time in clinical trials? I'll go back to BAN 2401 adaptive trial for Alzheimer's. This is now, the treatment is lecanemab. It was the first disease-modifying treatment approved for Alzheimer's. This is Eisai, ran the development of this. They ran a phase 2 trial, and in the phase 2 trial, they had 5 doses and control. The doses were different dose levels and frequency, monthly or bi-monthly infusions. And they ran an adaptive trial that included response adaptive randomization over the different doses. Whichever doses were working better, they wanted to put more patients on that to learn what is the right dose going forward. They wanted proof of concept. If the predictive probability of winning phase 3 got high enough, they wanted to jump to phase 3. They could stop for futility. The trial had an adaptive sample size from 300 to 800. The primary endpoint in the trial was 18 months in that. Now, to make all these decisions in the trial, it's hard to do a response adaptive randomization if you have to wait for patients to get to 18 months. Now, the primary endpoint in that trial was ADCOMS, which is a cognitive measure. It's actually a combination of different questions from CDR sum of boxes, ADAS-Cog, and MMSE, and they were selected to be earlier in the disease course, cognitive type endpoints that would be more informative for early Alzheimer's disease, which is what was done in this trial. In order to be an efficient design, we needed to learn faster than 18 months. We get 3, 6, 9, 12, 15, 18 months values. Every 3 months, we get patients' data. All of that data was used to forecast the effect of the different doses relative to control, estimate the control rate, to speed that learning up, to have all of those adaptations take place. The prediction, so we use the same endpoint observed at earlier time periods. They're not primary, they're being used as an auxiliary endpoint, a predictive endpoint for the final cognitive outcome for a patient, which is 18 months. Now, in this trial, we did largely, the early part of the trial was predicting 12 months, but the full exposure went out to 18. So I'm going to alter that a little bit and talk about 12 months. The adaptive algorithm was driven by the 12 months. Now, the model we used was a linear regression model between 3 months and 12 months. 6 months and 12 months, 9 months and 12 months, informed by previous patients that had passed through the trial, passed through those endpoints in the trial itself. And we had previous data on the expected correlation of those. We used prior distributions for those correlations from previous data, but then that's updated in the trial itself on that correlation to then drive the estimates of the treatment. It's a beautiful example, in part because Eisai published the results of every one of the interim analyses. You can go in and see what did it forecast at the first interim analysis of the trial, which was 200 patients. And then every 50 patients in the trial, it did an update. At that first interim analysis, which is triggering 12 months, there were 0 patients at 12 months. At the second interim analysis at 250, I think we had several that had reached 12 months. A good number have gotten to 9 months, which is really predictive of 12 months, and 6 months. So they're progressing through that. Response adaptive randomization started at that 200. At that time, the response adaptive randomization immediately wanted the 2 high doses, 10 monthly and 10 bi-monthly, and kind of went away from the lower doses based on cognitive values at mostly 3 and 6 months. The randomization probabilities change, then the data get updated 50 patients later. And the exposure of the early patients gets longer, which is critical in adaptive design because that's the most valuable data in the trial. The values were updated again. Interestingly, in the trial, the final sample sizes of this, by the time everybody got to 18 months, were about 250 patients on the control, placebo, about 250 patients on 10 monthly, and about 175 on 10 bi-monthly. If you add together the other 3 doses, it was about 175. So these 2 doses got the lion's share of patients, appropriately so. The 10 bi-monthly arm eventually is the arm that went to the phase 3 trial and demonstrated superiority. About a 27% slowing of disease. Interestingly, in the adaptive trial of 800 patients, if you go through the movie of what happened, that forecast was about interim 5 or 6 or 7, right in there. The algorithm started to estimate a 25 to 30% slowing and probability of benefit above 95%. The probability of at least a 25% slowing, which is what the criteria was in the trial, that was what they wanted to define go, at interim 5 was about 75%. 75% chance that you have at least a 25% slowing. The final number, when all patients got through 18 months, years later, was 76%. Now, there was the huge value of the follow-up and all of that, but the algorithm was forecasting that. Now it's forecasting it by doses and placing patients on the right doses. And it made for a much more efficient trial design. It enabled them to explore 5 doses by the ability to do response adaptive randomization. But none of that happens without the longitudinal modeling of the endpoint. You learn nothing until it's too late to make those adaptations. Okay. A couple other examples of this just to give you the wide view of what this looks like, different trial designs utilizing this. The Trulicity is Eli Lilly's, it was a GLP-1, kind of kicked off Eli Lilly's jumping into GLP-1s, which I assume everybody understands the story of the impact of these. This was treatment of diabetes, and they ran a seamless 2-3 trial, the Award 5 trial. So you can go see the publications of that. And in the phase 2 part of it, they were exploring 7 different doses of dulaglutide. The trade name became Trulicity after it was eventually approved. The 7 different doses were explored. The primary endpoint was HbA1c. Interestingly, there was a utility function over 4 endpoints where they wanted to make the decision on allocating the doses and dose selection for phase 3, taking into account HbA1c change, blood pressure, and heart rate. So they didn't want those things to be elevated to the point of cardiovascular risk. The fourth endpoint was weight loss. The more weight loss, the more benefit it was perceived, the better the therapeutic profile and the better patients would want to take the treatment. If there was weight gain, there was no commercial aspect of this treatment. Ironically, that became the huge part of GLP-1s are now approved specifically for weight loss, even in non-diabetics. And that played a huge role in that trial. That was one of 4 endpoints in a utility function. Over the 7 endpoints, response adaptive randomization was done, the ability to jump from phase 2 to phase 3. And it was predicting, will it show non-inferiority to sitagliptin as an active comparator for the primary endpoint of the eventual move to phase 3. It made that's a 12-month endpoint, and during the trial we're enrolling and we're getting monthly values on HbA1c change, and we built a model between those values in 12 months. It jumped to phase 3. It selected 2 doses, 1.5 milligram and 0.75 milligrams, to go to phase 3, and there were no patients at 12 months. It was entirely based on the longitudinal modeling of HbA1c change, which was pretty well understood, the behavior of that endpoint. They had previous data on patients, and again, we spent a lot of time building that model. Interestingly, early on, the question was, what's a better predictor of 12-month HbA1c? Early values of HbA1c or fasting blood glucose, which is a measure that is thought to move faster, and maybe that's a better predictor. We actually had data to jump in and look at if we were going to predict 12 months, which one was better, and HbA1c was better. It's thought that that's kind of an integrated value over time, and maybe it's slow moving, but it was much better predictor of 12 months than fasting blood glucose was. And so these decisions in the building of that trial all go in because we're trying to accelerate it. Now, Lilly did something tremendous in that trial, and I've not seen it in another trial, is they knew that this longitudinal model was a really important part to selecting a dose. That dose that went to phase 3 spawned cardiovascular trials, other trials. That was the decision on dose. It was making that drug development decision. The 1.5 milligram dose became the dose that was used for dulaglutide and has made billions for Eli Lilly. And so that key decision, they didn't want to enroll too fast. Remember, this whole thing is about time to information. How quickly do we learn to make good decisions, like a seamless decision to go to phase 3? They controlled enrollment. We simulated the trial for a range of enrollments, and we saw that if you enroll too fast, you pile patients in, it's less efficient than enrolling slower to allow for further follow-up and data. We were making better decisions. It led to a more efficient development strategy to go slower. And they were worried if they handed it over that operationally we want to enroll fast. They knew that slower was actually more efficient of a development strategy. And they controlled it, and it went there. It jumped to phase 3 as soon as it could. The data were amazing for the treatment, and it showed superiority to sitagliptin on HbA1c change. And they say it's accelerated development 12 to 18 months. The key to that was the longitudinal modeling of HbA1c and weight loss, the prediction of 6-month weight loss based on earlier values, which is very, very predictive, by the way. Okay, so also a really nice example of that. We do a lot of stroke trials. IceCap was the topic of a podcast last week, where the endpoint is 90-day modified Rankin. It's an ordinal scale 0 through 6 of their status at 90 days. In the IceCap trial, we use their 30-day modified Rankin status as a predictor of 90 days, and it's an incredibly good predictor. Interestingly, by the way, if you go back to the I-SPY2 example, I know I'm jumping around here a bit, the prediction of the MRIs, what was really interesting is the most predictive thing is if the reduction wasn't very great, if it was less than a 50% reduction, at 1 month, that wasn't too bad. But if it was a 25 or 30% reduction, very unlikely you're going to be a PCR. PCR is a high hurdle. If you weren't seeing 50% plus reduction at the first month and 90 plus at the third, you were unlikely. If you were seeing 90 to 95, it was kind of 50-50 whether you're going to get there. If you're seeing 99% plus, then you were highly likely. Some of the most predictive part in that trial was the lack of response, the lack of good MRIs became very, very predictive, where other values are less predictive, but valuable, that it's not negative. In MRS, for example, a 30-day MRS of 6 means the patient's dead is a really strong predictor of death at 90 days. Some of the middling values can bounce around a little bit, but it's incredibly predictive of 90 days. You can accelerate your adaptive design 2 months by using 30-day values. Now, how do you do this? Well, we incorporate Bayesian models, not surprisingly, within it. They're really good for longitudinal models because they incorporate uncertainty really, really well. They can use prior models, prior estimates of that correlation. So you're doing analyses with little data early on. You want it to be smart in that circumstance. So what we do is we build a model and we have a distribution of the parameters of the longitudinal model. That's updated through Bayesian modeling, Markov chain Monte Carlo. And then we make a forecast of every patient that's early in the trial. Based on that longitudinal model makes a forecast. We multiply impute those values, which gives you a complete set of data. Every patient has a final value for that. Then we update the final analysis modeling because we have a complete trial. And then we do that again and again and again. So at every interim, we're simulating 10,000 complete trials where that imputation is done through the longitudinal modeling. Become a very common technique now for missing data. Very valuable technique for missing data. That's the similar stuff we're doing for an adaptive trial in the middle of it. So the fun part of this, we do a lot of this with different kinds of endpoints, different kinds of predictors. Within the trial, we get to build these models from them, this ordinal model of 30 days to 90 days. If you're a 3, how do we predict values at 90 days? If you're a 4, how do we predict values? Do we want those forecasts only to look at the patients that were 3 at the early time point? Or do we want a monotonic model like in I-SPY2? There was a monotonic model that the more reduction, the better. So you're learning about all of the values from neighboring ones as well, potentially. If you're a 2 at 30 days, do we want forecasts to be better, which is a better value, than a 3? So do we want that, or do we want to only look at patients that are on that value? You might even have non-monotonic forecasts. These are all the things that go into building this adaptive design machinery that, by the way, when the trial's over, the machinery goes away, unless you're using it for missing data within the circumstances of it. We've actually now done a number of stroke trials where we're using 24-hour NIHSS, NIH stroke score, as forecasting 90 days, and it's actually really good forecasting. It's really, really good. And then updating it based on 7-day status, 30-day status. to 90-day status. Again, accelerating the learning to make a better trial design. Okay, number of other examples of this, wide range of types of variables, wide range of potential outcome variables, spending a lot of time on what those models look like, how do we make inferences, what do we know going in, prior distributions, all in the name of accelerating learning to make a more efficient adaptive trial, treat patients better, make better development decisions, enroll patients in a more efficient way, making a better design through this mechanism. All right, appreciate you joining me here today. Appreciate you joining me in the fight against time. Sometimes we're trying to slow time. We're all trying to slow time. Here, we're trying to speed up time. We want to improve learning, speed up learning through longitudinal modeling. All right, I hope you can make your adaptive trials more efficient. And until next time, we'll be here in the interim.